I'd like to introduce Allison Okamura, another member of the Tsai Executive Committee, to chair the first session.
All right, thank you everyone for coming. Is this working? I am pleased to introduce the speakers for the first session. Just to give everyone a heads up on timing, we'll have about 30 minutes for each talk and then 10 minutes for Q&A. So please think of your questions during the talk. And we really encourage trainees to ask questions as well. So the first speaker in this session is Larry Abbott. He is the William Bloor Professor of Theoretical Neuroscience and Professor of Physiology and Cellular Biophysics in the Biological Sciences. He is a principal investigator at Columbia's Zuckerman Institute and is co-director of Columbia's Kavli Institute for Brain Science, of course, all at Columbia University. And Larry, we're looking forward to your talk. Thank you.
Maybe I'll go to it. Thank you. This is a gigantic topic, as I'm sure you all know, and I'm going to start on the ground floor and take you up maybe a millimeter or so, and then leave it to the other speakers to soar into the stratosphere of cognition and all that. But I am going to address a question that's very important to me, which really concerns modeling. That's what I do. I model neural systems. And, you know, normally what a modeler does is to take the information you have on the left side there, build a model, and then put out some information about how a neural circuit affects the behavior of an animal, how its outputs, what it's going to look like. And in the kind of modeling that I'm going to talk about, which is fly circuitry, the input consists of measurements, physiology, but also connectomic data, so data about the detailed connections of the circuit. And so this is what we sort of do, and through most of my career, the only person who's done it are humans, but that obviously has changed. That's what today is all about. And now, you know, modeling, whoops, let me go back for a second. Modeling can be done, of course, very well by machines as well. So I want to talk about the contrast and a comparison. It's sort of a fight, to tell you the truth, between human model building and machine model building. But the problem in doing that in this this setup, I got ahead of myself. In this setup is that the right side, which is sort of the judgment of the model, can be vague. Neuroscience is hard, behavior is hard. So it's hard to know whether the machine got a better answer than you did, a human did. So I got ahead of myself, but what I'm going to do is flip it around, because connectomics is something very definite. And so I'm going to build models, given the function of a circuit, given the physiology, and try to predict the connectome. We'll do it with humans, and we'll do it with machines, and we'll see how things work out. Okay, so on the human side, in the left-hand corner of the ring, are the humans. And the humans predominantly for my talk are my collaborators, Gabby Mayman and Peter Muscles-Pires, but there's a lot of assistance here. So this is the human team that I've assembled for the talk. Included here is Shal Druckmann, who's I don't know here, but at Stanford, who worked with Rachel Wilson. Anyway, a lot of smart people. Now on the other corner of the ring is my laptop. So you might think I've rigged things. You know, we don't have 40,000 GPUs over there. It's just this thing. And indeed, I thought when I started preparing this talk that I would tell you how much better humans are than machines. But you'll see it didn't quite work out that way, despite the fact that I rigged it by having all these smart people on this side and not a very impressive computer on the other side. Okay. Now, what we need, of course, to decide who's doing better is the data. And the data in this case comes from Peter in part, and also from this very beautiful paper on the connectomics of the fly central complex from the Jayaraman lab. Okay, so here's the function that we're going to talk about, the task, if you want. It's a simple task. As I said, this is going to be simple. And the point of it being simple is partly that I do simple things, but also that we want to know, we want to be able to say, is the answer right or wrong? Now, whether I'm going to do that or not, you'll see. But we need to start on things we understand well. And this is a circuit that we understand quite well. Okay, so here's the idea. A fly is moving along in the world. It knows which way it's heading. I'll talk to you in a little bit about how it knows that. And it has a goal in mind. And so it's heading towards its goal, but then for some reason it gets blown off course, okay? And it has to figure out, I have to turn to the left to get back to the course and make that turn. So I'm going to talk about the circuitry that does that, compares the goal to the heading and generates a steering signal that turns the fly back in the right direction. Okay, so I have to introduce you to the circuitry here. This is done by an area of the fly brain called the central complex because it's in the middle. This little donut thing is called the ellipsoid body. That's part of it. That's where the heading signal comes from, as you'll see in a second. Between the circuitry I'm going to talk about ultimately and that ellipsoid body is something called the protocerebral bridge, this kind of arch. And then the main computer here is this fan-shaped body. That's kind of the navigational computer of the fly's brain. Okay, now the top object is obviously a circle. It's literally a donut-shaped object in the fly's brain. And it can be, parameterized by an angle going around the circle from 0 to 306 degrees. That's very natural. What you can't see from this diagram, but you would see if I showed you the connections, the connectomics of this circuit, is that these are really all circles. The two, kind of handlebars of the protocerebral bridge, you should think of them as being closed off into a circle. And so they can also be parameterized by 0 to 360 degrees. And if activity goes off the right side of one of those circles, it just pops back, or one of the handlebars. Likewise, the fan-shaped body is like if you took a fan and you turned it into a circle. That's the way you should really think of it. Physically, it's not that, but as far as a circuit goes, it also can be parameterized by angles. So you're going to hear a ton about angles in this talk because the whole structure is about angles. Okay, so the heading of the fly, what we need is heading and goal, right? And then compare them and generating a steering signal. Heading the fly, as Selig and Jarama showed, is encoded in the fly's brain by a bump of activity, a hot spot of activity within the ellipsoid body, like I've shown with the blue dot. And what happens is that encodes heading because if the fly turns, that bump moves in the fly's head and counter-rotates. And sort of like if I point my hand towards the door and then I turn around on the stage, but I keep that orientation. So that's the fly's heading. That signal gets sent down to the protocerebral bridge. And now I'm going to draw another person here, Chung Liu, who's here and at Stanford, who I collaborated with along with Gabby. And again, Shal Druckmann and Rachel Wilson did work on this. For the nature of how this signal, the heading signal, gets transmitted down to the computation that's going to do the steering. Because we need to know, basically what I'm trying to do from a machine learning standpoint is say, what's the input to this circuit so that I can run the machine learning? Okay. And the answer is that bump of activity gets turned into a sinusoidal pattern of activity across these structures. Remember, they really are circles, even though anatomically they don't look like it. So you can think of now a sine wave of activity around the circle. And when the fly turns, that sine wave goes back and forth, slides back and forth. And so the angle that's encoded at the top, kind of trivially as the angle around the circle, becomes the phase of those sine waves. There's a one-to-one correspondence. So that's how heading is going to enter into this system. Okay, now we get the fan-shaped body because that's where the steering signal is going to be computed. And here we call on Peter's data because what Peter found when he was doing his experiments is the goal signal. Remember, we need a heading signal and we need a goal signal to figure out how to turn. And that goal signal is also a sinusoidal pattern of activity, but in this case across the structure of the fan-shaped body. And so again, that peak of the sine wave, the phase, would be the particular goal angle for the fly at this moment. So now what happens, if the fly has a goal, wants to fly out that door over there, but gets blown off course, what's going to happen is it will record that its heading changed, but the goal won't change, right? As long as the fly's goal is still to fly out the door, and then maybe it gets blown back and forth, but ultimately the steering signal brings all those into alignment again. That's the idea here. Okay, so now we get to the crux of the story. Given this setup, so we know how the heading is encoded in the fly brain, we know how the goal is, how do you combine them and compute a steering signal? Okay, well that is done by these neurons. This is a family of neurons that, as you can see, tile the fan-shaped body. They also go to a region called the lateral accessory lobe. You see that best on the left side here, which is really the right side of the fly brain. And they're going to do this job. And they do this job by getting the heading signal and the goal signal. Now, just so you can get oriented, I just drew a schematic there because the angle's a little different. But you should be able to see the protocerebral bridge arches there and the fan-shaped body. They're just a little bit overlaid in this picture. So just to get oriented. Okay, so they get the heading signal up in the fan-shaped body, and they get the goal signal up in the protocerebral bridge, and then they get the goal signal in the fan-shaped body, and then they deliver their output, and that's the steering signal, to these two lateral accessory lobes, which are premotor structures, connect to command neurons that tell the fly to turn left or right. And what we're going to assume here is that the difference between the left-going signal and the right-going signal is the ultimate turning signal. Okay. So the question is, how do these inputs come in? And then we can start building models. Once we know the in and the out, we can build a model. So I'm going to not show you the whole set, but just take one of these neurons individually. There are 24 of them in all, what you saw in the previous picture, but now let's look at one of them. And again, it gets its heading signal through the processes in the protocerebral bridge there. It gets the goal signal there, delivers the output on the side there. And I'm going to call the heading angle H and the goal angle G, very simple. And I already told you then that there are these sinusoidal patterns of activity across these structures. So if this happens to be cell I, 1 out of the 24, what we'd expect for its input is that it would be something like a cosine, would be exactly actually a cosine, of the heading signal minus, depending on where it picks off the cosine, right, it's going to have what we call a preferred heading signal. So that's the heading signal where it would respond the most if the heading was in that direction. So we expect the input to look like that. Now, I could put an amplitude factor there, and I could have done the whole analysis that way, but I've simplified it and just assumed all the amplitude factors are 1. You'll see that'll come back up again. Okay, the goal signals are very the same. You have a cosine, a pattern of activity. We just have to say, well, what does that cell pick up? Well, what it picks up depends on where its preferred goal is. Where does it want the goal to be to respond the most? But it's exactly the same. And if you just put these together, you get a conclusion that the responses of these PFL3 cells, which are the steering cells, should be some function of a bias term, just an offset, a term that describes their response to the heading, and a term that replies their response to the goal. And the parameters here are the bias. Let me just go back because I got ahead of myself again. Sorry. The bias, and I'm going to simplify the problem by assuming they all have the same bias. Again, I didn't have to do that, but it simplifies things. They have a heading preference, that's what we have to do, and they have a goal preference. So if we know all those numbers, we can just put this all together and run the thing, and we should get the right answer. Now, I've simplified matters because I'm going to give both the human and the computer the preferred goal angles. We kind of know those, and I'll just assume those. I'm going to give them the bias that was measured experimentally. So the whole problem is to determine those angles. That's the machine learning problem. That's the human problem. Okay, now one thing if you know machine learning is you need to know this function. You need to know the nonlinearity. Fortunately, Peter measured it. And it's a function actually quite familiar to machine learning people. It's called a soft plus function. So we know the nonlinearity. We'll give that to the human. We'll give that to the machine. And again, the object now is to say, fit the steering output of this circuit and then tell me what those preferred headings angles are. And then I'm going to look up in the connectome and figure out what the right answer is, and I will grade you on that, both the machine and the human. Okay, there are 24 of these neurons. That means there are 24 of these angles. There are 24 parameters, but they happen to be angles. And so that's what we're going to be talking about, the values of those 24 numbers. I said this is not, this is not large language models. It's a very small model. But it's a case where we can look to see how the both human and the machine are doing. And then, what both the human and the machine are going to have to do is get those angles so that when you take these neurons or these responses and you add them up on the left and the right, there are subsets. 12 of these 24 project to the left side, 12 project to the right side. Add that up, take the difference, and you should get the right turning signal. All right, just one more thing I have to do. What is the right turning signal? The right turning signal has the shape of a sine wave, okay? It's measured by Peter. This is Peter's measurement. It isn't as simple as one curve. There's one curve like this for every goal direction. So it's really fitting an infinite number of curves. But they can be collectively expressed as it should be the sine of the difference between the goal and the heading. That's a function that's 0 if you're headed right towards your goal. It has one side if you're to the left, the other side if you're to the right. That's the steering signal. All right, so that's the problem. Here's the answer. This is the connectome that carries the presynaptic heading signals into these neurons. And the nature of those connections are going to determine these preferred heading signals. Okay? So it's a big matrix. It looks like that. But we're going to simplify it. Again, I didn't have to do this, but it makes it a little simpler. When we simplify it, because these presynaptic inputs come from the protocerebral bridge, and we have this angular label, so we can identify them with angles. And that's what I'm going to do. So I'm going to synthesize this big matrix in terms of a set of 24 angles. The other thing about this matrix is some of these cells are kind of duplicates of each other. It's not really as big as it looks. Okay? So I'm going to take this data, and that's really the answer that I'm talking about. These are the preferred heading angles predicted by a very naive, raw analysis of the connectome. I've depicted them obviously as angles around a circle. There are 12 dots on the left. Those are the angles of the 12 the PFL3, these steering neurons that go to the left side. There are 12 dots on the right. Okay, for now, that's the answer. Although humans, never accept anything. The humans are going to fight this a little bit. You'll see it's coming. Okay, so here's the machine learning problem. I'm going to start with the machine learning. You have 24 units in the middle layer, and I've remapped it a little bit, but this is exactly what the machine learning problem becomes. You give it signals that are the cosine of the heading and the sine of the heading. You give it other signals that are the cosine of the goal direction and the sine of the goal direction. You feed that into this layer. You read out an output from this layer. The answer should be sine of g minus h. That's the machine learning task. You can all program it while you're sitting there on your laptop, and you can solve it while you're sitting there on your laptop. Okay. However, we're going to be very generous. We're going to give it those connections. We're going to give it those connections. The only thing that machine learning has to do is get those connections. And from those connections, we'll extract these angles, and we'll see how the machine did. So just to get back to this amplitude issue, if you think of the two connections from the input to any single unit, that's like a two-dimensional vector. It's two numbers. What I'm doing here is I'm assuming it's a unit vector. And so I'm going to characterize those two numbers by single angle. That's the way I've simplified it. Okay, so here's the thing running on my laptop. It goes like this. These are sample of the possible curves that it has to get, and it does fine. So it works, okay? It shouldn't surprise you. This is a very simple task, and machine learning is good. So there's the AI solution. You get a beautiful sinusoidal curve. And this is another way I'm going to show the results. This is saying that if you're, where does the steering signal go to zero, basically? Where does the steering system tell you to go? That should match the goal angle perfectly. And so this should be a diagonal line. If you notice, it has a little wiggle to it, but it's pretty good. So the machine learning has done a good job. And it got these angles. It got essentially what looked like random angles. So this is very machine learning-esque. Namely, machine learning doesn't care about neatness or anything like that. Just makes it work. This set of angles makes it work. It's not unique. I could have showed you other set of angles that makes it work. Okay? So that's kind of where we are, but we're not done. And we're not done because Peter measured more than this. I mentioned that this turning signal is actually the difference of two signals. And he measured those two signals on the either side of the animal, on the right and the left. And so we can look and see how did machine learning do on those signals. Here they are. And it did terribly. Okay? Again, very machine learning-like. We as humans would always say we're modeling an animal, we're going to make it left, right, symmetric, reflection symmetric, as animals all are. The machine learning doesn't care about this. It's gotten this weird solution where one side is hardly doing anything and the other side's doing all the work. Doesn't match it at all. All right, so you got to be a little bit more instructive to the machine. And that is, we're going to put in extra outputs, and we're going to say, get the left and the right signal correct as well. Okay? You've got to constrain a little bit more. It's a very unconstrained problem. When you do that, of course, the machine learning does it. It's great. And it does even better on the angular accuracy of the turn. And it gets a set of angles. Now, I'm only showing you single examples. I'm not doing a statistical analysis. You'll have to trust me, which I'm a human. It's a bad idea. But here it is. And what you notice, if you'd run this a bunch of times, is the angles are now a little more symmetric than they used to be. By constraining the system to have left-right symmetry, it's also constrained the angles to be a little more uniform around the circle. Okay. Now, there's another thing about this circuit that we need to get right. This comes from large-scale brain imaging, something that Tom Clendenin has done here at Stanford and gotten this result. This happens to be from Evan Schaefer. What you find if you look over the whole brain is that there's a huge modulation by the speed that the animal's traveling, whether it's still, whether it's walking quickly, walking slowly, essentially a simplification, but the whole brain's activity goes up and down, kind of all over the place as a function of activity. And what you can think of very simply as what's happening is we have these sinusoidal inputs, and they're kind of going up and down all the time. So what happens if you do that to the machine? Well, it doesn't do too well. If you have these up and down vibrations, now the goal signal is kind of wiggling around rather than staying on the diagonal, the steering signal rather, so it's not matching the goal. Now again, you would say, well, you didn't tell the machine to do that. Okay, so we go back and we say, let's add noise to the machine and see how it does. And it does quite well. If you'll notice, although it's worth noticing here, I'll come back to this, there's still tiny little wiggles, but it pretty much suppressed the noise, okay? Did well. And it found an even more symmetric pattern of angles, okay? It's being pushed in the symmetric direction. But at this point, I think the machine declares victory, okay? And it declares victory by showing you this graph, okay? This is just one example of the machine. It was not taken to match the bottom. This is just one thing I randomly took from the machine, but it matches incredibly well, right? And of course, you can do more statistical analysis. It finds kind of the right range between perfect symmetry and complete randomness that's in the ballpark of what the connectome is telling you. And this is what I said. I didn't think this would work. A week ago, I wouldn't have thought it worked, but I did this for this talk. And it works amazingly well. Okay, so the machine declares victory. Time for the human, okay? And okay. What does a human do? I think this is true of all modelers. The human in this case is me, but I'm going to represent the species. And I think, in a fair way. So what does a human do? Again, assuming you don't know anything, you just need to put out this thing. Human is going to produce a very symmetric, neat, efficient solution, okay? This is what the human is going to pick for the angles. And in fact, this is what I did. When I started working with Peter, this is exactly the construction I made. And he immediately wrote back and said, you're wrong. And you'll see why he did that. But okay, that's the human. Beautiful structure, very neat and elegant. It works, okay? There's no fitting, right? That's just it. You stick it in and you get the answer. And it gives you a nice sinusoidal fit. It gives you an absolutely perfect alignment of the steering, the zero of the steering with the goal. And this is really because it's symmetric, right? But by using symmetry, there are all sorts of restrictions on what the answer can be, and it works beautifully. But interestingly, if you look at the same thing that the machine got wrong, the human gets it wrong too. Namely, the two side curves are much too symmetric, right? The human overdid the symmetry. And they're not, they are just perfect copies of the middle signal with a sign flip on the left. And that's wrong. However, humans, are clever, and all you have to do to fix that, you don't even have to change the angles, you just have to reassign them. You notice in these plots, I didn't tell you which of the neurons has which angle, and if you just do a little reassignment, then boom, you get it right, okay? So humans doing well here. And an advantage of using symmetry is that you get this for free, okay? Now you put in this up-down motion of the signal. You haven't even thought about it previously as the human, and it has absolutely no effect. There aren't even any little wiggles here. It has no effect. And again, that's because of the symmetry. So, there's something to be said for being a human. Okay, so, but now you say, well, is that a good approximation of that? And now, it's going to get into the mushiness of my response, my conclusions, okay? So the human could say, well, the fly is just sort of a sloppy version of my perfectly symmetric solution, whereas the machine can say it's a sloppy version of my sloppy solution. And so we have a fight on our hands. And here's the difference, okay? Here's the human prediction against the data. Here's the machine prediction. You can decide which one's better. But I'm going to stand up for humans myself. So let me do a little more. First thing the human says, humans don't go down easy, right? The first thing a human notices is that occasionally the machine comes up with a symmetric solution. Okay? Just occasionally, but the machine knows that sometimes works. The other thing that the human might notice is, and you might notice, is that there are a lot of pairs in the data. The bottom is the data. It's looking like it's paired up. And so now the human's going to do something in the tried and true tradition of humanity, is fiddle with the data. So first, let me show you what happens. If you take these data, namely those angles that I just showed you, and you build the system, so this is now not a model, this is just straight from the data, here's what you get. Okay, again, no parameter fitting, just boom, that's what you get. Now, the human's going to quibble with this, but you got to admit, this is pretty amazing, right? This is a zero parameter model. It's a zero parameter construction of a real neural circuit, and it works amazingly well. But the human's going to quibble, so, it's not super accurate. It has a little wobble. Now, for all we know, flies also are not super accurate in this, and it doesn't resist the noise that I talked about. So it's not perfect. And so now the human's going to fiddle with the data, okay? And this is actually something we did right from the start, Peter and Gabby and me, which we kind of cleaned up the connectome a little bit. What we did is we looked for the sort of dominant paths in this structure, and we got rid of the rest. So we did this. You can take a look at what we did. We just cleaned it up a little bit. Not with any kind of malice of forethought. We didn't know what this would do, but we did it. And when you do this, it has an amazing consequence. So you can say, what I've been showing you is the angles that come out of analyzing that matrix. But what if you look at the angles that characterize that matrix? They're perfectly symmetric. These are them. No, there's no fudge. You just stick in the numbers, boom, that's what you get. Now, if you're still awake, you might notice that there's only 8 dots, and there's supposed to be 12 on each side. That's because it's an unusual setup where some of the angles get doubled up. In other words, some of the angles are used twice. It's a kind of a weird arrangement, but it's highly symmetric, and this works extraordinarily well. If you just, again, let the data build its own model, it fits this beautifully, it gives a perfect thing, and it's completely resistant to noise, again, because of the symmetry. So, perfect. So, the human's going to say, that's the answer for me. And although I got the symmetry wrong, I had the right idea. Okay? The human imposed the maximum symmetry that the system can have, which is a 12-fold 30-degree symmetry. What the fly does, if you believe me, is a 4-fold 90-degree symmetry, but it's still a symmetry. So here's the final score sheet. And I don't have a conclusion. I say, we should do it both ways. The machine did a pretty good job of reconstructing these angles, and amazingly good, in my opinion. The human, however, led us to a possibility that the fly system is actually constructed on a symmetry. And I have to say, I'm human, so what can I have to believe this. I actually believe that this is the truth. I think that the insect central complex is built on this basic symmetry. Individual flies may not realize the symmetry perfectly, but if you went, for example, to one of the insects that are much better navigators than flies, I would predict that you would get that symmetry expressed, almost perfectly. Okay, so that's my conclusion. I'll stop there, and thank you for being human.
Thanks. We'll now take some questions. I'm going to start over here. Please stand up, and if you can say your name as well, that would be helpful.
Daniel Cap, very exciting work. Are the fly wings symmetrical in strength? Is there a left side or the right side? Because if there is, then you wouldn't expect the symmetry.
I mean, I still would, because I think those would be corrected downstream, right? This steering signal doesn't go directly to the legs. So compensating for a little bit different size leg or a little different size wing on the two sides, I would say is probably done by the motor system. And I would still expect this to be symmetric. Because after all, this is generic, right? Different flies are going to have different asymmetries. So
damage a wing considerable.
I would say that this wouldn't change, but downstream would change. But I'm speculating.
Thank you. Next we have Kang.
Kang Shen from Tsai. So you fudged with the data. You basically made, probably those data are noise. So you threw out those noisy data. So what if you fed the machine with the clean data? Would they come up with the same model?
Yeah. So yes. The answer is yes. I won't go on. Yes, if you want, exactly. If this was too hard to do the calculation, very good. Yeah, I didn't say that. So you could clean up the data, give it to the machine, and it would give you the answer that you wanted. So you can still fudge data even if you use a machine. I guess that's what I'm saying.
Great. Can we get any questions from trainees? All right. If not, you had to. Oh, got one right there. Okay. Let's pass over the microphone. Thanks.
You saw many times the ablation of going from the signal to then the only signal to then the two signals and then the noise. Yeah. When you invert this order and then you just, without doing the two signals, just add the noise, what's the behavior?
Yeah, I never did that, but I think, let's say if you did the machine, you added the noise, it would still have this weird left-right thing, because there's no pressure for it to do a left-right symmetric solution. So I don't think you would have got a different answer, but I didn't do it that way.
Even with the pressure to correct for, because you're not training for correcting that wiggle.
Because you can correct for that by symmetrizing the angles. I don't know. I should try it. I didn't do it, so I'll try it. As I say, it only takes like 30 seconds. So.
Great. And the next question is from Scott in the back.
Yeah. Hi, Scott Delp. I'm from here at Stanford. Thank you. That was a interesting talk. I'm wondering about your underlying assumption that the difference is what matters. If you build a robot that has to steer and you only feed it the difference between the goal and your heading, it doesn't work very well. It'll wobble around. You need to take the derivative of the difference, and then it works really well. So I'm wondering why it was just the difference, or if you needed a velocity feedback to help.
Yeah. There's certainly a velocity effects in this circuit. This is done with calcium imaging, so faster signals were probably missing. So I was reproducing the data, but it's entirely possible that there's some velocity stuff in there for quick turns and stuff like that.
Okay, thank you. Jay.
Hi, Jay McClellan. Nice to see you, Larry. I was just wondering if there was any possibility that there's a functional argument for why we get organized like this. And I guess Scott's question is moving in that same direction, but maybe you get a little bit of extra stability by lining these things up a little bit near the axes or something like that.
Yeah, so, another thing you can do, and actually I did this. So if you take away, I normalized the vectors. If you don't normalize the vectors, it's a much harder problem. Then the machine finds the symmetric solution, not the weird symmetry, same as the human. So, and this always happens. You can say if the machine doesn't give you kind of what you want, you haven't constrained the task enough, and I think that's entirely possible. But you don't get the right answer from the machine. You get the human symmetric answer if you push like that.
No, but I was just saying that the data may be capturing a functional.
Yeah, I think they are. And this system will work better, right? The symmetric one. The other thing is that I thought you were mentioning about development. It probably has to do with the way the circuit develops, and it's sort of easier to specify a symmetric pattern than a non-symmetric pattern. So I think that's the other reason you could think that underlying the developmental plan is that, and then maybe it doesn't get realized totally perfectly.
Great. No, a question answered? All right. Others? Right there. Thanks.
Chung is the man who found the sine waves.
Thank you. This is Cheng from Stanford. I have a question, which is how bad comparing the bottom right data on the flight behavior versus the ideal situation? Because the reason I asked, because now you have shown two optimization process. One is human, one is computer. But there's another optimization process, which is evolution, that didn't make it to the bottom left, but stays at bottom right, which could be due to many other constraints. So how good is it for the bottom right?
Yeah. So the bottom right is really the biggest thing is that noise effect, that velocity. And, as you pointed out, there could be other effects that correct the velocity. So I wouldn't want to claim that I really know from behavior which one's right.
Okay. I think we have time for maybe
Bill?
one more. Sure, Bill.
Hey, Larry. Bill Newsom, Stanford. What is the physical interpretation of the doubling up on the data on the bottom left there?
It's bizarre.
Is there redundancy emerging in some of the signaling neurons? And does it make any predictions about, the effect of lesions that you could actually test this kind of thing?
Yeah. I don't think it's a doubling up for that purpose, like you only do it for every other one. When I saw this pattern, first of all, I didn't recognize it as symmetric right away, and then it just drove me completely crazy, but I've come to love it. I don't know. I don't think it's a backup sort of plan. I haven't sort of done lesions, but my suspicion is if you knock out one there or you knock out one on the right, it would equally screw things up. So I don't think it's a backup plan.
All right, we'll stop there. Thank you very much, Larry.